Hi guys. Okay, here we go. So we are finally to what a lot of people consider their favorite part of biology, and that is genetics and heredity and genes and all that kind of stuff. Um, and it'll take a little bit before we get to the genetics part, so you'll have to get through this part first, but I think it's interesting, so hopefully you will too. And so this is chapter 8. In chapter 8, we will actually divide into two parts. So this is chapter 8A, and this is going to be genes and cell division, mitosis, meiosis, those kinds of things. And then we'll get to the actual genetics. So here we go. So first things first, this is all about how cells within organisms, how they help the living organisms grow, how they help them reproduce. And so this, we're going to start out with what we call species characteristics. So every member of a species have characteristics that are common to all of its members. So for example, you could say humans have 2 arms and 2 legs. Humans have, they stand upright. Humans have hair on their head, those kinds of things. And so these are very specific to species. So if I have a person and a dog and a dolphin and all these different, and a plant, then I look at each of them and say, okay, you're a person, you're a dog, you're, and I do that based on species characteristics. Now, within a species, each creature, each organism has distinguishing characteristics. And so humans, again, have distinguishing characteristics like different color of eyes, or different hair texture, or different skin tone, or different height, or whatever it is, different voice patterns. And so in a classroom full of students like you guys, I can look at you and know who you are individually, and those are your individual characteristics. Now, those characteristics, species and individual characteristics, come from the genes that we get from our parents. So our biological moms and dads have contributed their genes, and that's where our characteristics come from. We are a combination of our mom and our dad. And so that's why you have some of you look very similar to one parent or the other, but you act like the other person, or you have the same, like you can't have lactose and neither can your mom, or, whatever it is. We get those characteristics from our parents. Who we are also depends on how we were raised, even before we were born, on the nutrition that was received, on like intellectual stimulus. Did you read a lot, or you read a lot too as a kid? All these different kinds of things go into developing who you are. And probably the most important is we are also very dependent on the development of our spiritual nature. Have we been taught right from wrong? Have we been given more than just do this, do that, but also given the reasons and who we are obeying, and those kinds of things. And so there's a lot that goes into it. Scientifically, we will go back to the genes because those are what are actually studied. But be aware that it's not all genes. Okay, so now heredity. Genetics is the study of heredity. Back in the very old, early days, this was studied simply because of breeding livestock. And so you want a cow to produce a calf that will grow up to be big and strong. So you have a lot of meat on it. You have a lot to eat. And so there have been records since recorded time of like stock records. It's a new science because we now have the ability to truly look at the molecules and kind of figure out what is going on at the molecular level. We don't know it all, but we have a pretty good start on it. And this has been a long time coming. We have always been interested as people in how we pass on characteristics. I guarantee you will not go to the hospital to see a newly born baby without somebody saying, oh, he has his mom's nose, or, oh, she looks like her dad, or, something like that. And so it's always been a thing. And it started from the beginning. The ancient Greeks, of course, had a theory. Remember, they have a theory on everything. And they believed that children were a mixture of elements from their parents' bodies. Aristotle thought it was actually something in the parents' blood that mixed together to form offspring. We talked a little bit in a previous chapter about the idea of preformationism. This was the Victorian era, 17th century. Women weren't that important, and that basically the offspring came from the sperm, which contained a tiny little completely formed organism, and that grew into an adult eventually if it was planted into an egg. So there's, our little picture of the little tiny person curled up. Now, the real beginning of genetics started in the mid-1800s with a man named Gregor Mendel, and we will talk a lot more about him as we get to the second part of this chapter. But he performed a series of genetic breedings and took extensive records. And he was a gardener. He was a monk, and anyway, he was the gardener, and he bred these garden peas and looked at their different characteristics and came up with the idea that he called factors. And those we now call genes, but they are the genetic information that parents give to their offspring. He did an amazing job for the time, and modern genetics is actually based on his experiments. And then, jump ahead, I guess, about 100 years, and we had Watson and Crick figure out the shape of a DNA molecule, which became very big because it just gave us the next step into studying the molecular levels of DNA and how they change. And of course, hopefully you recognize that picture going from a chromosome to being kind of unraveled to getting to the double helix that we've already talked about, and then all the way down to the base pairs with the A's and the T's and the C's and the G's. That's where we're at. And so and everything that we are going to talk about now is dependent on that pattern. Okay, so these genes, an individual gene is a piece of DNA or a section of DNA that produces a very specific, particular polypeptide chain of amino acids, which is basically a protein, and that protein causes a particular trait. And it can be a visible structure, hair color, skin tone, whatever it is, or an internal structure, an enzyme that your body uses, or the way that your specific neurons work in your brain, all those kinds of things. And so all of those are determined by your genes, and the DNA contains all of a person's genes, and every cell in your body, and we've talked about this before, remember, whether it's in your eyeball or your big toe, every cell has all of the genes for your whole body. And so only certain parts of the DNA are used in certain parts, but every cell has this information. And there's my little fun fact down there. If you take out all of a person's DNA, it's about 60 grams, which is very, very small amount. But if you stretch it all out, it would go to the sun and back about 100 times. Who figured that out? I don't know. If it's super, actually true, I don't know, but it's kind of cool. Okay, chromosomes. So here we go with these chromosomes. So first off, each DNA strand in the nucleus, and depending on the organism, you have different numbers of chromosomes, but each DNA strand in the nucleus contains these genes, and these long strands of DNA are called chromosomes. And in a non-dividing cell, so a cell just kind of living its life, the chromosomes are kind of fuzzy, kind of tangled up. And at that point, they are called chromatin material. So, and I apologize in advance for science being dumb because a lot of these words are going to sound similar. But so, just in general, cell's doing its thing, then the DNA is called chromatin material. Now, within that strand of DNA, within those chromosomes, you do have one very important support structure, and those are called histones. Histones are there to kind of keep the DNA kind of under control. And so they help support it when it's kind of uncoiled and keep it, even though it looks all tangled and looks kind of fuzzy, they kind of keep everything kind of a little organized. And then when it does coil up, which we'll talk about in a minute, the histone helps to coil it and pack it, and then helps to kind of keep the shape of that chromosome. As far as we are concerned, and what we will talk about as we look at the chromosomes, they will most often resemble an X. And this is what is seen when a cell is actually dividing, and I'll show you some pictures of those. And in that X, each half of the X is what is called a chromatid. Sorry, I know that's dumb, chromatin and chromatid. And they are two identical parts. So if you have the X, it's like this. Here's one chromatid and here's the other. Okay, and together they make an X. So that's what they're talking about, half of the chromosome or half of the X. They are held together, so the middle part right there, that is the centromere. Okay, so now there are two types of chromosomes, or two kinds. First are the sex chromosomes, and these tell you whether an organism is male or female. They don't have a lot of other information on them. Besides the gender of an organism. For humans, the sex chromosomes are X and Y, so very specific. The X chromosome does have more information on it, and each gender, male or female, does have at least one X, so that information does get passed on, does get used. And so if you are female, then your chromosomes will be XX. You got one X from mom and one X from dad, and you are a girl. If you are male, you got one X from mom and you got a Y from dad, and you are male. Okay, that is, those are the two genders. I know there's a lot of issue with that in today's world, but you're either a boy or a girl, and your genetics prove it. So then, autosomes, these are the other chromosomes in the body. These are everything else. So these are hair color, height, skin tone, the proteins in your stomach, the enzymes that you use to do different things. All of the rest goes back to the autosomes. Now, one species characteristic that is very important and that can be used to actually determine what an organism is, if all you have is some DNA, is the number of chromosomes. Humans have 46 chromosomes in the nucleus of most of their cells. In all of their body cells, they have 46 chromosomes. In an egg cell or a sperm cell, they only have 23, which we'll talk about more. But so 46 is the number for humans. Fruit flies, just FYI, have 8. They're actually one of the most studied organisms genetically because they have such a small number. And then kind of random to me, crawfish, like a little tiny lobster looking thing, have 200 chromosomes. So that is not an indication of, how advanced or how smart or how, whatever an organism is, but it is a species characteristic. And one way that science has used to look at chromosomes is a karyotype. So when a cell is going through division and the chromosomes have condensed down to form their X's, then pictures are taken and they are arranged to show the number of chromosomes, any problems, those kinds of things. And they're arranged in pairs according to their length, because they will always be the same size, and where their I have chromosome location, and where their centrosome is located. Centrosome. And so with this picture over here, so notice, that some of them are quite big and the centrosome is right in the middle, and some of them are small and the centrosome, or where they're attached, is kind of closer to one end than the other. Notice the very last pair of chromosomes are the sex chromosomes. This is an X and a Y, so this is the karyotype of a boy. Okay, so karyotypes show the pairs of chromosomes, and like I said, humans have 46 chromosomes, 23 pairs. It's 46, or 23 times 2 is 46, and they are known as homologous pairs of chromosomes. And the homologous is because the two members of that pair contain the same information. Each member is known as a homolog. So again, I know a lot of the words sound the same. And cells that do have these homologous pairs are called diploid. And so you are considered a diploid organism because you have pairs, homologous pairs. And most organisms are diploid. We'll talk about a few that aren't, but for the most part, you have two pairs of chromosomes from each parent. So our diploid number is 46, like I've said. And in those homologs, in the pairs, they have genes for the same characteristics. So we have two of most genes. Again, there's some differences there, but you got a gene for hair color from your mom and from your dad. And sometimes you just see one or the other, and you carry the gene for the other hair color, but it's not there. It could be passed on, but you don't see it. Sometimes the genes, you'll see an expression of both of those characteristics. So you might have hair color that's kind of in between, but you have genes from both your mom and dad for every characteristic that you have, even if you don't show that characteristic. Now, some cells do only have one set of chromosomes. They don't come in pairs. These are known as haploids. In humans, the only haploid cells are sperm or sex cells because you are giving up half of your chromosomes to your offspring. Okay, so now this whole cell division thing. So we're going to start with the cell cycle. And so this is what your cell does in its life. It goes through these three major phases: interphase, mitosis, and cytokinesis. And so each phase has kind of its own part in all of this. And so this picture just shows everything, and we'll go through each little step. Okay, so here is interphase. This picture that you see on the screen, that is some onion cells. And they are, so I'll show you pretty much the same types of cells each time, but this is interphase. This is where cells are most of the time in their life. Cells grow during this time. Cells do their normal activities. They produce whatever they need to produce. They go through photosynthesis if they're a plant cell. They go through cellular respiration to get energy. They're doing everything all the time. But they also are going through these three parts of interphase. They're going through the G1 phase, which the G stands for growth. And this is between cell division and DNA replication. And so the cell's growing in size, it's reproducing the number of organelles, it's preparing essentially to divide. The S phase is important. This is where DNA is replicated, and each single chromosome is duplicated. So back a couple chapters ago, when we talked about DNA replication, this is where that happens, where everything in the DNA is copied and it goes through the whole process. And you, after this phase is over, have two sets of DNA in a cell. And then you go through the G2 phase, so another growth phase. So the cell produces proteins, produces molecules to go into the next phase of the cell cycle, which is mitosis. At the end of this phase, the cell is now a mother cell. So the cell is basically ready to give birth, to produce a new cell. Okay, so now mitosis. This is the actual division of the nuclear material, which is DNA, so that each new nucleus has a complete and identical set of genetic information. So now remember, we've already got identical sets of DNA. Now we just have to separate them, get them into two different nuclei. So mitosis happens in 4 phases. The first one is prophase. So here's a little picture of prophase. So notice, so I'm focused in on one cell, and so it looks a little different than before. You can start to see some lines in the cell. The the chromosomes are becoming more condensed. So they're getting out of that tangled kind of fuzzy stage, and they are condensing, getting shorter and thicker. This is so that they're easier to move around. So it's easier to pick up a ball of yarn than it is to pick up just a long kind of random strand of yarn. Okay, so in prophase, the chromosomes get short and thick, they condense. They become visible as sister chromatids, and then they're attached at their centromere, so that you see the two sides. Okay, so one for mom, so the pairs of each other. So now and then proteins come in and they attach to the centromere, to the middle of the X, and they attach it to what is called the kinetochore, and this is a fiber network. It's going to pull things apart. The nuclear membrane disappears, so you can't really see it on this picture anymore because it's pretty much gone. In general, you can kind of see like a circle around the nucleus. The centrosomes are outside the nucleus. They're usually going to end up over like on two different parts of the cell. And they form the mitotic spindle, which is actually going to start to pull the chromosomes apart once they get where they should be. Okay, so now the next two phases, we have metaphase, and in metaphase, the chromosomes line up on the equatorial plane. So they line up down the middle of the cell. So I know they look kind of random still, but they line up right down the middle of the cell. This is the easiest one to recognize. And then anaphase. The sister chromatids separate, and now they are back to being individual chromosomes. So when they were together like this, they were sister chromatids, and when they separate, they become individual chromosomes. And so what happens is the little pulleys, the little fibers that are attached, and then basically kind of reel them in and pull them apart. Okay, then we have telophase, and telophase begins when those chromosomes get to the opposite sides of the cell. The nuclear membrane will begin to form around each set, so it's going to start making a new nucleus, and the chromosomes begin to get uncoiled. They start to look fuzzy and kind of tangled up again. Okay, so that was mitosis. And now cytokinesis. This is the final step. This is the division of everything else. So all of the cytoplasmic content. So the cytoplasm itself, the organelles, the proteins, all that stuff. And depending on a plant or an animal, this happens a little bit differently. Plants form a cell plate. And so this, I'll kind of draw it in a little bit more. So this cell plate is formed by different vesicles right between the two nuclei, and basically splits that mother cell in half. Eventually, the plant will add reinforcements to this plate, and it will become a cell wall. Now, in animals, so I have a picture down here that's a whitefish set of cells going through mitosis. They don't have cell walls, remember, and so the cells tend to be a little rounder. And so what happens is filaments form like a little contractile ring that will divide the cell. So it's kind of like a drawstring, so like on a bag, and then you pull it tight and it kind of squeezes in. So this causes the mother cell to kind of go from a circle and kind of pinches in. So you have two separate cells, and then they can separate, and it forms what they call a cleavage furrow. So that's kind of this little indentation here and up here. So that's forming, separating those two cells out. And once the membrane pinches enough together, then your two cells are formed. Okay, so cells are going through this all the time. If cells are growing, or if the organism is growing, that happens through mitosis. If a cell is damaged or is so damaged that it needs to be fixed, then not a cell, excuse me, an organism, then it can be to produce replacement or repair cells. Some organisms do use this for asexual reproduction, which basically is just producing like a clone. Fragmentation, you see this with things like sea coral or sea sponges, where a piece of the organism just breaks off and a new complete organism will grow from that. Budding, organisms like yeast or certain bacteria produce buds. So just like a new cell grows from the old one. Spores, lots of molds are distributed through spores, and so it is usually you see it in much simpler living organisms, but it is a pretty common technique for reproduction. Okay, so now meiosis. This is where we get haploid cells. This is where we get like an egg or a sperm cell. And so you need haploid cells because two haploid cells from the two parents will unite and form a diploid cell called a zygote. So every single one of us started out life as a zygote. That was a union of an egg and a sperm. And so to get those, the cells will go, the sex cells will go through meiosis. Excuse me, and this is actually the reduction of a cell's chromosome number from diploid, so in our case from 46 to haploid 23 by two consecutive cell divisions, meiosis 1, meiosis 2. All right, so we're going to go through these steps again. So basically we'll go through like the prophase, metaphase, anaphase stuff, and just kind of talk about some of the differences, what is different here versus mitosis. So first things first, the cells go through interphase. The cells grow, the DNA replicates. So again, you have twice as much DNA as a normal cell. So if you had 46 to start with chromosomes, we've got 92 right now. So chromosomes coil up in prophase 1, they condense, they become visible, all those kinds of things. The mitotic spindle forms, so this is going to be the pulley system. And then in this one, the homologous chromosomes pair up. So remember that you have a chromosome for everything from mom and a chromosome for everything from dad, for all your different things. So say that the blue chromosome on the screen is from your dad, the green chromosome is from your mom, and the two parts are because it's been replicated. So you have two of everything. And in this phase, now they not only line up the X's, but now you have the homologous pair line up also. So these chromosomes have the information for your hair color and your hair texture. And so you have everything about your hair from mom and everything about your hair from dad, and you have both sets copied. So you have 4 pieces of information about your hair texture and color. Now, very often during this phase, which they call synapsis, when you have the two X's lined up together, and that actually is called a tetrad when you have the two X's. During this phase, during synapsis, when the chromatids are close together, sometimes the genetic information gets transferred from one chromatid to another, and this is called crossing over. So notice that the little legs of these two chromosomes basically switch places. So now you have a chromosome, this first one, so we'll number them. That first one is exactly like what your dad gave you about your hair color and your hair texture. But this second chromosome, so this part or this piece, it is mostly what your dad gave you with a little bit of your mom. And then the third piece is mostly what your mom gave you with a little bit of your dad. That's a 3. I can't get it drawn on there. And then the last one, this fourth little piece, is exactly like what your mom gave you. This is why you sometimes will see kids have identical features to one parent or the other, if you got, the first or the fourth chromosome. But it's also why very often you will see a kid who has a combination of what the parents gave them. And so this crossing over leads to some genetic kind of mix-up, and not in a bad way, but in a way that makes each person unique with different characteristics than their parents. Okay, so then metaphase 2, or excuse me, metaphase 1, the tetrads line up. And so this picture down at the bottom, so it's got the tetrads, it points out crossing over potentially. And then you have the metaphase, so the chromosomes are lined up. The tetrads, the two X's, the pairs of X's are lined up in the middle of the cell. In anaphase, the X's, the pairs of X's now get separate, and so you have homologous pairs that get separated from each other, and they move to the ends of the spindle, so they go from being close together to far apart. And then you have telophase 1 and also some cytokinesis. So the chromosomes get to the new cell or away from each other. So they arrive at the poles, which is just basically the opposite sides of the cells. They don't uncoil yet. They stay X's, but the cells do split. And so you get two new cells, and those cells immediately go into meiosis 2. So you have two cells going through this at once. And so again, prophase 2, the mitotic spindle reforms. Metaphase 2, the chromosomes line up on the equatorial place. And remember at this point, each cell, basically, if we're talking about people, has 46 chromosomes. So everything lines up. In anaphase 2, the sister chromatids are separated. So 46 gets divided in 2. And so now each cell will have 23. And because this is happening with two different cells, you actually end up with 4 new cells being formed, and each cell is a haploid. Okay, each cell is has half the number of chromosomes that an organism needs, because it needs to combine with either an egg or a sperm to produce the offspring. Now this final little picture shows kind of the whole steps all together. So I know it's small and you can't really see what the X's are doing or what is happening, but I want you to just kind of see it as one whole thing. And notice that you have a repeat of the phases. You go through 2 series of basically the same steps. We'll talk about some of these other things that are listed on here, like the segregation, the independent assortment, all that kind of stuff, as we get into more of the genetics. But I do want you to see the difference between mitosis, which is what body cells are doing, and meiosis, which are what your sex cells are doing. Okay, so as always, if you are listening to this for the first time, we'll go over, of course, in class, and I'll probably do a better job and answer questions, obviously. If you're going through it as a review, hopefully it will help kind of go back over some of the things that you have learned. As always, feel free to ask me questions. Watch it however many times you want to, if you need to, and I'll see you later. Thanks, guys. Bye.